In my group, we are interested in understanding the evolutionary basis that explain why organisms age in different ways. There are organisms that are extremely long-lived. For example, some sharks live centuries. Other organisms that are very short-lived. And also at the mechanistic level, at the molecular level, we want to understand what factors make them be so short-lived or long-lived. So at the same time, we are also interested in the interactions between organisms and their environment, and how this crosstalk between, for example, the microbial environment that surrounds organisms and the organisms themselves contributes to the aging process and to health in general.
There's millions of microbes living in your gut, like millions and millions, and that's a good thing, right? So they help you process food, they help the immune system function properly. But we know that in patients that have Alzheimer's or Parkinson's disease, for instance, so neurodegenerative disorders, that these patients have a disturbed microbial community, so there's a disbalance. Some species take over the system. And it is thought that this could actually cause the brain to degenerate. But at this point, that's just a hypothesis. So I'm trying with killifish to help answer that question. And killifish is a great model for that because they have naturally, they both have a degenerating brain as they age, and they have this balance in microbes as we see in Parkinson's and Alzheimer's patients. I'm doing these experiments to find out whether indeed microbes can cause brain degeneration. And the way that I go about it is that I'm trying to make the microbes in the gut again get balanced. So I treat fish with antibiotics, so I get rid of their disbalanced microbes, and then I give them microbes from young fish that are still in balance, so I restore the balance. And then I investigate whether these fish still have their brains degenerate or not. And the way that I measure that is either through behavioral testing, so we know that young fish learn very well, and old fish learn much less well, just like in Alzheimer's patients, for instance. They have decreased learning capacity. We also see that in these fish. And maybe by targeting the microbiota, we can help them maintain their learning capacity into old age. And the other way of testing it is to extract the brain from the treated fish and then look for molecular markers. So, for instance, if we see neurons that are degenerating, as we would also see in Alzheimer's and Parkinson's patients.
In our fieldwork, so briefly, we work in Zimbabwe, in Southeast Africa, in the Gonareju National Park. We study in this particular part of the world the natural history and ecology of the turquoise killifish. And so the reason why we keep going there is because we want to understand the evolution of the turquoise killifish in its natural environment. We explore this type of question of what's the evolutionary and demographic ultimate cause of current observed aging-related conditions, not just in fish or humans, but we also study them in a more theoretical, in a more kind of like a numerical approach. So we run simulations to actually abstract and to generalize general laws of how organisms evolve to be long-lived or short-lived or to age fast or slow. I really strongly believe in diversity in science at multiple levels. So thematic diversity and background diversity and the skills diversity. The concept of diversity for us is actually central even to the research that we do. Actually, we study microbial diversity as a hallmark of a healthy microbiome. We study actually immune antibody diversity as a hallmark of richness and health and resilience and robustness of host immune system, for example. And so we study kind of like this ecological diversity as a research topic, but then very much embrace that also as a team.